Welcome back to my home. I'm out in my yard on a kind of a muggy gray day. It's been raining, so the air feels a little bit like it might start raining on me at any moment now, so we'll see what happens. But let's keep going. So we're here today to talk about neurotransmitters. So this reflects our second core concept in the field of neuroscience. Again, continuing on in our thinking about the important means by which neurons communicate using electrical and chemical signals. So now we're going to focus on what exactly those chemical signals are. So our learning objectives today are that I want you to be able to name the major small molecule neurotransmitters and neuropeptides in the central nervous system and briefly state the functions of each. And I want you to think with me about the factors that determine the effect of neurotransmitters on postsynaptic neurons. All right. Well, let's begin by reviewing what we talked about last time about the organization of chemical synaptic transmission. And this slide gives you again an overview of the processes that are involved in chemical synaptic transmission, beginning with the steady state, where we find in the presynaptic terminal a variety of organelles, and of course our presynaptic vesicles that are loaded up with neurotransmitter ready to go. And what happens then with the arrival of an action potential is a wave of depolarization spreads along the pre-terminal membrane, which eventually reaches voltage-gated calcium channels. And those channels open, calcium rushes in, which is the key triggering event, which leads to the fusion of the presynaptic terminal membrane and the membrane of the synaptic vesicle. So that happens as the snare complex, which is present out here, twists and tugs and pulls those membranes together once calcium associates with synaptotagmin. And as those membranes begin to fuse, they form what we call a fusion pore, and that allows for neurotransmitters then to diffuse out of the synaptic vesicle into the synaptic cleft. And from there, the neurotransmitters can associate with receptors for those neurotransmitters in the postsynaptic side of the synapse. And as receptors and transmitters interact, typically ion channels open and close. That could lead to the generation of a wave of depolarization in that postsynaptic process, and maybe even with enough summation of inputs that are doing the same thing at the same time, there could be an action potential generated in that postsynaptic cell. Meanwhile, the membrane of the vesicle is recycled back for further activity. And this involves the coating of the vesicle membrane with a protein called clathrin and the association of clathrin with other molecules that facilitate the pinching off of that vesicle membrane, and then it's trafficking back into the pool of vesicles that are ready to be filled again with their neurotransmitter and re-enter this vesicle cycle. Okay, well, let's let that suffice as a brief review of chemical synaptic transmission. Again, I would refer you to the animation associated with the textbook Animation 5.2 on chemical synaptic transmission, which will review all of these processes in a nice, short, succinct presentation. All right, well, what we're here to talk about today is neurotransmitters. And neurotransmitters come in two broad kinds of categories based on their structure. And these two categories of neurotransmitters interact with different kinds of receptors. So we have two broad classes of neurotransmitters based on the size of the molecules. One category we call small molecule neurotransmitters for obvious reasons. And then to contrast those small molecule transmitters, we have neurotransmitters that are actually small peptides. And so these peptides, of course, are comprised of amino acids that are covalently bound to one another, and sometimes these peptides can be quite sizable. So small molecule transmitters and peptide transmitters. So these two classes of transmitter bind to two classes of receptor. One class of receptor we call ionotropic receptors. These are receptors where the receptor site is actually part of an ion channel, and that ion channel can open or close when the transmitter binds to the receptor. So these ion channels are called ligand-gated ion channels, with the ligand being the neurotransmitter. The other type of receptor that we'll consider today are called metabotropic receptors, and these receptors activate second messenger system. So there are metabolic steps involved in mediating the effect of the neurotransmitter once it binds to its receptor. Now, our small molecule neurotransmitters can actually interact with both of these classes of receptors, both the inotropic receptors and the metabotropic receptors. So right away, I want to emphasize to you that if we want to understand the activity of a neurotransmitter, we need to know something about the receptors that it interacts with. And the activities of neurotransmitters, even on the very same cells, can be remarkably diverse, depending upon what kinds of receptors are expressed on that neuron. Our peptide neurotransmitters, as far as we know, as far as I know, only interact with metabotropic receptors. So, as we'll come to see, these metabotropic receptors mediate slower and potentially longer lasting effects in postsynaptic neurons. So we associate our small molecule transmitters primarily with quick actions, and our peptide transmitters, generally speaking, with more long-lasting and slowly evolving actions. Okay, well, let's begin with a survey of some important small molecule transmitters. Now, for those of you following along in the reading here in Chapter 6 goes into more depth than what we'll cover. So I want you to focus on the level of knowledge that I've given you in the tutorial notes and the level of my discussion with you all today. So let's begin by thinking about these small molecule transmitters and get a broad functional sense of what these molecules are. So many of them are amino acids. Some of the amino acid transmitters that are important in brain and spinal cord function are shown here. They include amino acids such as glutamate, potentially aspartate, certainly gamma-aminobutyric acid, or GABA for short, and glycine. So these molecules mediate rapid synaptic effects when they interact with their ionotropic receptors. As I mentioned, they may also interact with metabotropic receptors. So the small molecule transmitters can also have longer lasting effects when those particular kinds of receptors are bound. Now, here we're illustrating some of the important small molecule transmitters that you'll want to know about, and perhaps the most important of them all is glutamate. So glutamate is typically considered the major excitatory neurotransmitter in the central nervous system, and as you see here, it is an amino acid, so short carbon skeleton with the amino group attached to the side opposite the carboxyl group. So glutamate is involved in various aspects of cellular metabolism, but it also is concentrated in synaptic vesicles at the terminals of neurons that use glutamate as a neurotransmitter. Now, this is such an important molecule that we've prepared some animations that talk about the processing of this molecule as a neurotransmitter, so I'd encourage you to pause just a minute and view Animation 6.2 on the website that supports our textbook. You can navigate there yourself or click on the hyperlink in your tutorial handout. Well, next, let's consider acetylcholine. So we're deviating just a little bit from the flow of the handout, but that's okay. Acetylcholine is another small molecule transmitter.